how do we generate next-gen sequencing data? How do we really understand what is important in that data from a research perspective and then also from a clinical perspective, and how do we start integrating that with other validation technologies? In addition to that, I was also involved with the opportunity to develop and teach a course at the University of Minnesota, specifically around their Bachelor of Health Science program, and really had the opportunity to see how data is being used not just for research and precision medicine, but also for educational programs. And what I realized was it's all the same data. It's the same data being collected and used for all of these different applications. And so the real focus for me became, how do we build systems, how do we find solutions to access and use that data appropriately? And that's really the focus of my position now with Oracle. As a researcher, again, my main focus was in genomics, spanning the whole variety of genomics, DNA sequencing, RNA, microarray, a whole variety of validation data types such as qPCR and nanostring, also, Sanger sequencing, all of the standard bench science. So I was very much a bench science researcher. From there, though, we were starting to get involved with early stage clinical trials and starting to build tools that the clinicians were desperately asking for, starting to find biomarkers, and we started branching out into cell culture data, ELISA, flow cytometry. And what we realized is that a lot of these data types are very different. Some of them are highly structured, and some of them are less structured. Some of them, like flow cytometry, at that time it was rapidly evolving. When you start using it for things like immunophenotyping, you realize that the methods for analyzing and using that data really change pretty rapidly. And so you have the need to have really dynamic environments for storing and using that data. And then you layer on, in a translational research and precision medicine setting, the clinical data coming from EMRs and from other, outside publicly available sources, and the complexity continues to increase. The tools that I used primarily were things like, you still see a lot of Excel. Excel is very much the, workhorse of the research environment. And while it's a great tool, it does open you up to some disadvantages, which we'll discuss throughout the course of the webinar today. A lot of open source tools, that's really common in research and in academics, started to see more complex visualization tools coming in, and then also a variety of point solutions, so things like Galaxy, FlowJo for flow cytometry, Partec for some of the genomics work. And really the question was, how do you fit all of these point solutions together and use the same data? Some of the main challenges that we had to address, first of all, was access to data, access to samples. How do we figure out who has what data? Everyone has 10 hard drives, piled up above their desk in their lab. You have a postdoc leave your lab. Who has access to that data? Who knows where it is? And how do you share it? And when you are sharing data, how do you do that without violating policy, especially around any patient data or anything that has clinical information with it? So really just how do you integrate the different data types and share that and maintain all of the required policy by the institution? And specifically, I think this is a really important point, is the data sharing requirements. So for anything that's associated with patients, you have to have institutional review board approval, and you have to be sure that the data is only used by and shared with the appropriate number and level of people, and that the identification of that must follow policy. And that's a really difficult challenge when you have diverse teams where you may have some clinicians that can see all of the PHI-associated data, but then you have other members of the team, say a biostatistician, and the data has to be de-identified. And so that was always a challenge to make sure that you are maintaining that level of security around the data. You also have to track and document who you're sharing data with, where it's going, but also it's very difficult to transfer data especially large data sets. It's a little bit like that game of telephone where, the first person in a long string comes up with a sentence and then whispers it in the ear of and it gets passed down the line. And by the last person, it's very different from the first. Data can be like that as well. If each person that gets the data makes a small change, by the time it is finally finished, going through all of the partners in the collaboration, it can look very different from what it started. So, really became interested in the tool sets that you use to control this environment and move successful research forward.